This lecture focuses on the cells that are involved in the immune system, or the immune response. So the first thing I want to actually discuss is just to take a few steps back, in that when we talk about the blood is made up of two different components. There is the liquid portion of the blood that's referred to as the serum or the plasma. This is non-cellular components that include proteins, glucose, mineral ions, hormones, and other molecules. The cellular portion of the blood contains three broad categories of cells. There are the red blood cells, the white blood cells, and the platelets. Red blood cells, or erythrocytes, are the cells that are involved in the transport of oxygen. White blood cells, or leukocytes, are the cells involved in immunity and fighting an infection. And lastly, platelets, or thrombocytes, are involved in prevention of excess bleeding. So since we are focusing on the immune system and the immune response, we will discuss the leukocytes that are the types or category of cells known as white blood cells. These are again the cells that are involved in the immune response. Leukocytes are categorized by two broad designations. Cells can be either categorized as a myelocyte, and this includes macrophages, neutrophils, eosinophils, and basophils, or they can be categorized as lymphocytes, which include B cells, T cells, and natural killer cells. And we're going to look at each one of these cell types in a little more detail overall. Before we begin to discuss the specific cell types involved in the immune response, I want to talk a little bit about how cells are produced, specifically how blood cells are produced. The process is known as hematopoiesis, and this is simply put the formation of and development of red and white blood cells. The root hemato refers to blood, and the term poiesis means to produce. So hematopoiesis is the process of producing blood. Specifically, I want to talk a little bit about a cell type known as a hematopoietic stem cell, or HSC. This is a cell type from which all lineages of blood cells arise. In general terms, a stem cell is a cell that can differentiate into other cell types. All of the genetic material is present inside every single cell, so what makes a specific cell type specific compared to another cell type? For example, what makes a heart cell different from a liver cell? It is the genes that are being expressed in those specific cell types. Now, a stem cell can differentiate into any cell type. Other cells have their fate locked in to produce only specific cell types. So a hematopoietic stem cell is a type of stem cell that can produce any of the cell types that are involved in blood that we will discuss in just a minute. Now, a hematopoietic stem cell cannot become a liver cell or a heart cell. It can only produce blood cells, but it is still undifferentiated, meaning that it has the ability to become any of the blood cell types. Also in this slide, there is a graph that focuses on hematopoietic activity when, during the lifespan of an individual, specific components of the individual's physiology are involved in production of blood cells. So very early on in fetal development, the yolk sac is involved in allowing for blood cells to be produced. Then the transition occurs to the liver and the spleen. Again, this is all during fetal development, and this is where the majority of hematopoiesis takes place. There is then a transition before birth and right at birth where the long bones are involved in hematopoiesis. And in adults, we also know that the cranium, pelvis, sternum, ribs, and vertebra also allow for hematopoiesis to continue. This slide and the following slide are meant to give you an overview to get an understanding of how hematopoiesis actually takes place within a vertebrate system. In this particular slide at the very top, you see starting off is what's known as the hematopoietic stem cell. This is again a stem cell type that can differentiate into any of the blood cells. First of all, study of hematopoietic stem cells actually is very difficult. It turns out that these types of stem cells are actually very scarce. There is less than 1 in 50,000 hematopoietic stem cells found in the bone marrow. Because they are scarce, they are also very difficult to culture. So our understanding of the process is not complete. Even though we do have some information, the complete information is still a little lacking. I also want to introduce the concept of what's known as a progenitor cell. A progenitor cell has lost its capacity for self-renewal and is typically committed to a particular lineage. So again, at the top of the slide, you see a hematopoietic stem cell, which still has the ability to become any type of blood cell. At some point, there is a differentiation step that occurs where either you produce a myeloid progenitor or a lymphoid progenitor. Once this differentiation has occurred, those progenitor cells can only produce cells from the lineage from that point on. And for myeloid cells, that includes all of the cells listed here: dendritic cells, neutrophils, macrophages, eosinophils, basophils, platelets, and erythrocytes. So the myeloid progenitor can differentiate into erythrocytes as well as platelets. But more importantly, for our understanding of the immune system, we have those immune cells that are listed on the left-hand side. If the cell has differentiated into a lymphoid progenitor, this is still a cell that can produce other cell types, but the fate is locked in that only lymphoid cells can be produced. Lymphoid cells include natural killer cells, T cells, B cells, as well as dendritic cells. This figure is from a review article that also shows or exemplifies that differentiation process. There's a little more going on with this particular slide than the previous figure, but I think it's a really nice one to show all of the cells that do end up getting produced from a hematopoietic stem cell. So in this figure up at the very top, again, you see the hematopoietic stem cell. There is differentiation that occurs, but what I want to highlight, which is similar to the last slide, is that you have a myeloid progenitor or a lymphoid progenitor. In either one of these cases, the fate is now locked in for that lineage, myeloid lineage versus lymphoid lineage. And then from that point on, you can see all the cell types that can arise from either the myeloid lineage or the lymphoid lineage. Interestingly, at the very center of the slide, you see the dendritic cell. And if you noted on the last slide, dendritic cells are found in both the myeloid category and the lymphoid category. This particular cell type, which we will discuss in a little more detail, is an interesting cell type in that it can be derived from either a myeloid progenitor or a lymphoid progenitor. This is the only cell type that can be derived from either one of those progenitor cells. So let's break it down and look at it a little more simply. A majority of the myelocytes are also cell types that are considered phagocytes. So what I want to do is discuss the process of phagocytosis, which is a mechanism that then describes the cell type involved in the process. What I like to do then is take a step back and discuss a mechanism known as endocytosis. Endocytosis is a mechanism that occurs in lots of cell types that allows for larger materials or molecules to be taken into a cell, and the way that it is taken into the cell is through vesicles that bud inward from the plasma membrane. So if you look at the left-hand figure on this particular slide, the yellow represents the plasma membrane of a cell utilizing this mechanism of endocytosis. The green spheres are meant to be the molecules or components in question that the cell is trying to take from the exterior of the cell and bring it into the cell. What happens is divots or pits are formed in the plasma membrane where the molecule then is trapped, and eventually the membrane closes up on itself to produce a vesicle, which is a small membrane sac that is now inside the cell, and you can see that those green sphere molecules components are now inside the vesicle, inside the cell in question. Again, this process is a very common process for the uptake of large molecules or components into a cell. There are several specific mechanisms of endocytosis. Specifically, we're going to focus on phagocytosis. The word or root phagin means to eat, and this is a mechanism that essentially is like taking in large molecules to break them down overall. Now, with phagocytosis, you have the concept again of internalizing the external component. In this case, because this is a very clear mechanism involved in fighting off a pathogen, what you see on the right-hand side is a bacterium that is being taken in by the cell that's known as a phagocyte in this same process of pinching in of the plasma membrane and eventually forming a vesicle with the bacterium inside the vesicle, which is now inside the cell in question. That is referred to as a phagosome. So the vesicle that has the bacterium in it inside the cell is known as a phagosome. The next step is for the phagosome to fuse with a lysosome. Lysosomes are organelles that are found in just about every cell type. They contain enzymes that help lyse or break apart large molecules. So in this case, the phagosome that contains the bacterium that was taken in fuses with the lysosome, and the enzymes found within the lysosome begin to degrade or break down the bacterial pathogen. The structure at this point is referred to as a phagolysosome. Now, one of two things can happen at this point. The complete breakdown of the pathogen definitely occurs, and if we follow that all the way to the bottom of the slide, what we can see is those particles are going to be released, which will then be removed from the host system. Alternatively, phagocytosis can also allow for a direct link to the adaptive immune response. Now, I'm not going to go over that in much detail with this particular slide because I like to, again, focus on smaller batches of information, but just keep in mind that phagocytosis results in the breakdown of the pathogen, and either those pieces and parts that have been generated by the breakdown of the pathogen are released and removed from the host system, or they are utilized in a direct link to the adaptive immune response. Now, the first cell type that I want to discuss is what's known as a monocyte. Now, a monocyte is still a cell that can differentiate into a macrophage. So for our purposes, we're not even going to focus on the monocyte. We're going to talk about macrophages, which are on the next slide. So macrophages are categories of cells that are phagocytes. They do participate in extensive amounts of phagocytosis. They are also cells that are referred to as antigen-presenting cells, abbreviated APCs. These are cells that act as that link between the innate immune response and the adaptive immune response that I just discussed with phagocytosis 2 slides ago. Interestingly, for macrophages, there are specialized cells in different tissues. So throughout the body, there might be specific specialized cell types with their own nomenclature that are, in fact, acting as macrophages. So for our purposes, I like to generalize and call the macrophage a macrophage and not worry about the specific name of the cell type, if it's found in bone, or if it's found in the lung, or if it's found in the liver. So to simplify again, we will just call macrophages macrophages, and note that there are specialized cells found throughout the body that act as macrophages but have distinct nomenclature associated with them. Macrophages also secrete molecules known as cytokines. Now, this is a term that we have not introduced just yet, but a cytokine is a small signaling molecule that helps perpetuate the immune response. We will talk about cytokines extensively throughout the semester. For now, let's just leave it as that they are signaling molecules that help the immune response in some way. So macrophages have the ability to produce these signaling molecules, these cytokines, that help the immune response. Additionally, macrophages produce complement proteins. I mentioned the complement system very briefly in the first lecture, in that we have the innate immune response and the adaptive immune response, and then a third facet known as the complement system. The macrophages are involved in producing molecules, proteins involved in the complement system. Lastly, it turns out that macrophages can also produce what are known as reactive oxygen species and reactive nitrogen species. These are things that help with the destruction of the pathogen. They include things like superoxide, a hydroxyl radical, hydrogen peroxide, and again, these are all components that help with the breakdown of the pathogen overall. The next set of cells are all referred to as granulocytic cells or granulocytes. They are collectively called granulocytes because when you look at them under the microscope, they appear to have structures that look like granules inside. The granules all contain different components depending upon which cell type we're discussing, and I will focus on that as we discuss each cell type specifically. So the one that's on this particular slide is the neutrophil. The neutrophil is the most abundant leukocyte in the immune system. It makes up about 50 to 70% of leukocytes overall. It is in fact a phagocyte, meaning that it participates in extensive amounts of phagocytosis. It tends to be the very first type of cell to arrive at an inflammatory response. We will discuss inflammation in our next series of lectures. However, the neutrophil tends to be the first cell type to arrive at the site of an infection. This cell type can also produce reactive oxygen species and reactive nitrogen species to help with the destruction of the pathogen via phagocytosis. Neutrophils also produce molecules known as antimicrobial peptides. Antimicrobial peptides, also abbreviated AMPs, are molecules that are short stretches of amino acids that have the ability to play a role in the destruction of pathogens as well. We will look at antimicrobial peptides a few lectures down the line and look at them a little more detail. But for now, we can just say that neutrophils produce these small molecules that help with the destruction of pathogens as well. The main targets of the neutrophil are bacteria and fungi. It doesn't mean that neutrophils don't play a role in fighting off other pathogens, but the main targets are bacteria and fungi. Now, those granules that I mentioned for the neutrophils typically contain proteases, which is a general class of enzymes that break down proteins. The granules in the neutrophil also contain those antimicrobial peptides we just talked about. In addition, among other things, neutrophil granules contain histamine, and histamine is a molecule that allows for vasodilation to help with inflammation. Now, we haven't discussed inflammation and vasodilation just yet, so I'm going to ask you to hold on to the term histamine when we get to those components, and we will refer back to histamine and what it does when we discuss inflammation. For now, suffice it to say that the granules of neutrophils do in fact contain histamine that can be released. The next cell type I want to talk about is an eosinophil. Eosinophils are also phagocytes. They play a really large role in tumor destruction. Oftentimes, we don't necessarily think about cancer as being fought off by our immune systems, but at the end of the semester, we will discuss cancer and how the immune system plays a really big role. We will hear throughout the semester this concept of altered host cells or altered self cells. Those are typically cells that are infected with the virus or are a tumor cell. So again, tumors are something that are fought off by our immune system regularly, and eosinophils do in fact play a really big role with tumor destruction. Also, eosinophils are very big players in targeting parasitic infections. So when we discuss parasites a little more in detail, we will come back to the eosinophils and how eosinophils play an essential role in fighting off those parasites. The granules of eosinophils do in fact contain cytokines, again, those signaling molecules that allow for the immune response to continue. We also know that some eosinophils contain enzymes known as ribonucleases. A ribonuclease is a class of enzyme that degrades nucleic acids, and this is very important for fighting off viral components or viral pathogens. In addition, we know that the eosinophils play a role in producing reactive oxygen species and reactive nitrogen species. The next cell type that I want to discuss is a basophil. A basophil is in fact the least abundant leukocyte type, makes up less than 1% of the white blood cells found in a typical vertebrate system. This particular cell type is not a phagocyte. It is involved in secreting cytokines, those signaling molecules that we've just discussed. It also is a very key player in fighting off parasitic infections, in particular parasitic worms. And the basophil is involved in the hypersensitivity reaction, also known as an allergic response. Basophil granules contain cytokines, as well as histamine, as well as other molecules involved in the immune response. The last of the myelocytes that I want to discuss is a cell type known as a mast cell. Mast cells are also in very low abundance overall, similar to basophils. They act similarly to basophils. They are cells that allow for the production of histamine, as well as heparin, which is an anticoagulant. And again, we will discuss the importance of that in lectures to come. The second broad category of leukocytes are known as lymphocytes. Lymphocytes derive from a lymphoid progenitor cell, and ultimately the lymphoid progenitor can produce B cells, T cells, natural killer cells, as well as dendritic cells, again, like we mentioned in the previous slide. Lymphocytes tend to be the cell type that are key to the adaptive immune response. They allow for providing diversity, specificity, and memory in the adaptive immune response. Approximately 20 to 40% of the body's white blood cells circulating in the blood and the lymph are made up of lymphocytes. Even though the lymphocytes are lumped together as lymphocytes, the B cells and the T cells act in a similar way, and we'll discuss that here in just a minute. Natural killer cells and dendritic cells actually act in a very different way or a different mechanism or manner altogether. So T cells and B cells can also be subdivided and lumped together. Specifically, B cells and T cells have what are known as antigen receptors. So the term antigen is defined as any substance that binds to an antibody or a T cell receptor. An antibody is a receptor that is found on a B cell. So lymphocytes have the ability to interact with pathogen with these antigen receptors. And again, we'll discuss those in a little more detail in just a minute. So as I mentioned on the previous slide, both B cells and T cells behave in a specific way to allow for their activation to take place. This particular slide is focusing on a B cell, but the mechanism is exactly the same for a T cell. Both B cells and T cells come in what's known as a naive form, and that's what you see in the far right-hand corner. A naive lymphocyte, B cell or T cell, is a type of cell that has not yet interacted with any type of antigen. An antigen can be something that is part of a pathogen or secreted by a pathogen that can be recognized and interacted with one of these receptors. So again, a naive B cell or a T cell has not come in contact with any type of antigen just yet. When interaction with an antigen does in fact take place, that begins this very complex and orchestrated activation mechanism which allows for the production of specific cell types. The cell types that are produced via this activation include effector cells. Effector cells are the cell types that are going to do something or have some effect in the immune response. And the memory cells are those cells that, again, essentially don't do much during this response, but are the ones that are laying in waiting for this subsequent infection by that specific pathogen. So to reiterate, both B cells and T cells come in a naive form, which means they have never interacted with a pathogen, specifically the antigen of a pathogen. Once that interaction has taken place, the activation and the differentiation occurs of that cell to produce effector cells. And again, I'm broadly going to state that effector cells have some effect. They do something, and we'll talk about what that something is in just a minute. And the memory cells, which now have the ability to recognize that same antigen on subsequent exposure of that antigen. So let's talk about B cells in a little more detail. B cells are cell types that produce what is known as an antibody. Now, an antibody might be a term that you've heard previously. The term antibody refers to an antigen binding receptor. This is a receptor that can bind to an antigen. An antigen is any substance, usually foreign, that can bind to the antibody or the antigen binding receptor or a T cell receptor. So this concept sometimes confuses students in that B cells have membrane-associated antibodies as well as antibodies that can be released or secreted, known as soluble antibodies. The antibody structure, whether it is membrane-associated or soluble, is almost identical, like you see in the right-hand side of this particular slide. In the middle of the slide, you see the membrane-associated antibody versus the antibody that can be secreted. The membrane-associated antibody is typically involved in that first recognition step that we just talked about on the previous slide. Once that activation has occurred and effector cells have been produced, effector cells produce soluble antibody that can bind to that same antigen. It's soluble because it is secreted from the effector B cell, enters into the host bloodstream, and then tags potential pathogens for destruction. Now, we will look at B cell responses and the specific role of antibodies several lectures down the line when we get to the adaptive immune response. But for now, We will just talk about those soluble antibodies tagging pathogens for destruction. So effector cells that are produced after naive cells are activated are the ones that will produce soluble antibody. The next broad category of lymphocyte are the third type of lymphocyte that is distinct from B cells and T cells are natural killer cells. They are distinct from B cells and T cells because they do not have an antigen binding receptor, but their function overall is very similar to a cytotoxic T cell. Their job is to destroy altered self cells. They play a really big role in defense against viruses, fighting tumor formation by secreting cytotoxins, again, molecules that destroy altered self cells. So natural killer cells, hence the name killer in the title, play a role in killing self cells that need to be destroyed for one reason or another. Now again, they are distinct from B cells and T cells because they do not have that specific receptor that binds to antigen, but they are really important in the immune response overall. The last cell type that I want to discuss is known as a dendritic cell. A dendritic cell is in fact a phagocyte. It acts as an antigen-presenting cell, which we discussed previously are cells that play a role in phagocytosis and help with the connection between the innate immune response and the adaptive immune response. They do produce reactive oxygen species to help with the destruction of pathogens. They also do secrete cytokines as well as antimicrobial peptides. Now with dendritic cells, we have a similar situation compared to the macrophages. When we discussed macrophages, we said that macrophages have specialized cell types in the different tissues. Dendritic cells follow the same suit in that there are specialized cell types in different tissues. But for our purposes, we're going to lump dendritic cells together and just focus on dendritic cells as a generic term rather than looking at specialized names for cells in different tissues. So that completes the survey of the cell types that are involved in the immune response. And what we will do in our next lecture is look at the organs and organ systems that are involved in the immune response, and then we can jump into discussing innate immunity.